All right. Thank you, everyone. Welcome to week one. Hopefully you can see my screen. If you can't, probably show me a thumbs down. So we are off to a good start this week. And I had put a hit record there. And this week, we are going to introduce ourselves to the Terra platform, which is what we'll use throughout this training. And as a means of introducing you to the course, so the main aim of this week is to introduce you to the bioinformatics workflows that we use on Terra.bio and how these are used to analyze pathogen genomes for public health decision making. So you'll notice here there is that emphasis on the workflows that are used primarily for public health decision making. So again, giving you an overview for those who are not there last time, we have a Theiagen public health resources page, and here you'll find lots of resources describing the workflows, what each workflow does, what kind of inputs that we'll talk about that you need. We also have a link here on the slides that we'll share on the documentation for the Terra platform that we are using. And also we have the course training page that I shared earlier in the emails, but then I'll be happy to share again today. So should you need any help in the, yes, did I hear somebody? Okay. Should you need any help in the course of this training, please do send me an email. I'll be happy to help. And also just making you aware that there's more technical content that you can have if you get connected, if not done so already, with lots of pathogen genomics communities such as Phage, Staff B, or this Q Bioinfo Slack pages, for example. As a reminder, week one, this week, we are going to introduce you to Terra and how to undertake bacterial genomic analysis. And for the exercise, we'll import data into Terra and then run Theiaproc. For week two, we'll be looking at the individual steps that are part of the Theiaproc workflow. For example, the assembly, QC, and characterization. And the exercise will be looking at the outputs that were generated by the Theiaproc run that we'll undertake this week. And week three, we'll go into RC typing, where we generate phylogenetic trees based on a set of conserved ribosomal proteins. And the hands-on exercise will be generating this tree and looking at that tree. And week four, we'll go through phylogenetic trees, particularly in the context of public health, looking at methods that are used for phylogenetic inference as well as visualization. And then for the hands-on exercise, we generate these trees and visualize them using different tools. So this week we are driving straight in, and we hope that at the end of this week one, you'll understand what Terra is, understand how Terra works behind the scenes, be able to log into Terra, which most of you have done, import pathogen read data from NCBI onto Terra, upload pathogen read data onto Terra, and also run Theiaproc workflow on Terra. So first, we want to understand the Terra platform. So Terra is a bioinformatics web application, so it runs on the web rather than a local installation on your machine, running on an internet browser. And the benefit that this has is it's mostly point and click, and we think of it as an easier access to bioinformatic tools compared to somebody who has no background in coding, for example, or running things on the terminal. Here you're able to upload and organize your sequence data, as well as analyze that data with bioinformatics workflows. You can also access, download, and share results that you get from these analysis. So the web interface is app.terra.bio. So terra.bio is the main page. And then, when you click on this login button, it should be able to take you to the app.terra.bio. And we looked at this on Friday. Once you click on this 3 lines on the top left, you should be able to log in with your registered email, which in this case would be your Theiagen Cloud emails. And then once that is authenticated, you should be able to see the initial look will go onto the dashboard tab. But in this case, we want to look at or explore the different tabs that you'll be able to see when you log into Terra. Please feel free to stop me at any point if you have any questions. I'll be happy to help. So up here, the first thing you'll see is, workspaces. You'll see like who owns that specific account, and then you'll see the name of your workspace there. Then we have different tabs here: the dashboard, data analysis, workflows, and job history. The ones that we'll be interacting with are all of these except the analysis tab, which we don't often use, but can be used for other different types of analysis. So here in the data tab, this is where the data that you need to analyze as well as the results are organized, and it's accessible for viewing and or download. So here you'll see top here written table. So data in this on the Terra platform is organized in form of tables. And then at the bottom here, you'll also see that you have workspace data. Okay, I can see someone is requesting to share the link to the training. I will just copy that real quick and send that to them. Okay, so we've talked about the data tab, and then we also have the workflows tab. As you can see here, we have lots of tiles, each tile representing a different workflow. So these are the, this is where you'll find the list of the bioinformatics workflows that are available for performing analysis. We'll see how you can import workflows into your workflows tab for different kinds of analysis. And then we have the job history tab. So this is gives you a log of all the bioinformatic workflows that you've launched for analysis within the workspace. You'll see, for example, here we have the names of the workflows that were launched, the kind of tables there, the number of workflows, and also the status, which is quite critical. You'll see some of these, for example, are marked done and green. That's a good thing. But some of these are marked done, but with a red exclamation triangle, which means that they failed. And from here, you're also able to troubleshoot job failures, and we'll see how we can do that, because it's expected some of the analysis will run into issues and we'll have to troubleshoot. So I'll just do a quick demo going into this workspace here. Again, you remember the dashboard is this page that introduces, gives you a bit more information about the workspace. And here we just note that, these are some of the workflows that we've imported into your workspace. We've talked about the data tab. At this point, I still don't have any data tables here, but then we'll see how to get those there. And then looked at the workflows tab. At present, we have 2 workflows here. And in my submission history, I have not run its submission, so I don't have a log of the analysis that I have run. Okay, so I'll just go back to the to the slides here. So how does Terra platform work? So the interface that we see, like we've seen, we have data, we have those workflows tab, we have the job history. What happens behind the scene is like, you have Terra running on your browser, but at the back end, you have two critical components. So you have the bioinformatics workflows, as well as the cloud computing environment. So the bioinformatic workflows, think of it as a chain of different steps, where each step is involved in undertaking a specific task. So workflows are typically made of tasks. For example, you read in data, it checks for quality, it does assembly, another task does assembly here, maybe another one does alignment, and so on and so forth. And at the end, you get your output. So the bioinformatics tools, or some of these workflows that we are talking about, they're available on Docker. And the reason these are packaged on Docker is one typical problem is local installation is typically difficult. Disappeared for a second. Okay, got it back. So it's if you've ever installed bioinformatic tools on any computer, you will understand how slow sometimes that happens. Secondly, difficult installation. Sometimes you do have tools that you need to install fast before you actually install the tool that you need. And some of these can be difficult just based on the fact that probably you have different versions that collide or conflict, and you might not end up with the proper installation that you need. And one thing that is quite important in public health standardization is that when these tools or workflows are packaged in a Docker container, it ensures that everyone who downloads that Docker container uses the exact same version of the software. So wherever someone runs the same analysis on the same data, they should end up with the same outcome, because they're running the same version. So you don't end up with variations that might be due to different software versions handling the data differently. So the Theiagen workflows are written in a language called WDL, workflow development language. A short form, you should say WDL. And it's an easy language to write in and also understands. And again, going back to the principle of standardization, it ensures that the same analysis steps and the same parameters are undertaken for everyone. And it is also much faster. So, like we said, the workflows are hosted on Dockstore, and therefore you can pull these workflows from the Dockstore into your Terra workspace. And if you look within Dockstore, you'll see that different organizations, for example, Theiagen, that is hosting this training, along with other institutions, have several workflows hosted there. And I mean, just to reiterate this point, that your Terra workspace is not limited to only Theiagen genomics workflows. You can also put in workflows from other organizations as well. So the Theiagen workflows, so this is version 3.0.1, this just is a summary of the different workflows that Theiagen have developed. So, there are workflows for uploading data, fetching data, for example, from GenBank. We'll use one of these. You can also fetch assemblies, There's Theiaproc, which we'll use. There's a different one called Theiacov that is for viral pathogens. There's even Theiuc for eukaryotic pathogens, and there's even Theiameta for metagenomics. And then there are also workflows, for example, for phylogenetics like KSNP3, Augur, if you've used Augur before. So different workflows are taking different kinds of analysis. So when you run these workflows, you're using what we call Google Compute Resources, or in short, GCP. So again, going back to this image, you're running this Terra on your browser, and then you're actually using Google Cloud, and this is where the data is hosted. GCP provides, and also the not just storing the data, but as well as the computation resources to undertake this bioinformatic analysis. The data is stored in what we call GCP buckets. So such that when you click a link that you'll see for the files that are stored within these buckets, you should be able to actually download that data onto your local machine. So for example, this image you see here, it's telling you can download the data that in this case was clicked here for $0.01. I think this is just an estimated cost. Often it's way, way, way less than that. All right, so each workspace has one bucket where that data will be stored, and this data within that bucket can be accessed and analyzed by users with access to that specific workspace. And therefore, it means that another user's workspace cannot access the bucket from another user unless authorized. So something that we can look at later are the are the different access levels that users can have for their workspaces. For example, writer, you can write, you can perform analysis, or you can just be a reader, which means you can look at the data, but you can't launch a workflow, or you can be an owner of that workspace, which means you can do everything, including deleting of that specific workspace. As I mentioned, yes, with access, with permission, you can access data in other buckets. So this is something that gives you a bit more confidence that because people often fear the idea of their data being on the cloud. Your data can only be accessed by individuals that you've given access to. And this happens often because, typically labs would have like a specific workspace, and within that workspace, different members of the lab can have access to launch different workflows and undertake different analysis. So it can work quite nicely as a good collaborative environment. Now, we've talked about the data tab. So the data on Terra is actually organized within data table. And how you get your data onto this Terra data table is two-pronged. On one end, you can upload that data onto Terra. And two, when you launch a workflow, it will undertake an analysis, and the results will also be populated onto the Terra data table. There are two types of data tables. So sample level, and on a sample level, each row represents a single sample. And then we have set level data tables where each row represents a collection of samples. In the simplest form, if you're doing like, let's say you want to undertake genomic assembly, you'll be undertaking these on individual samples. But when you are undertaking phylogenetic analysis, you'll be doing this on a collection of samples. So you'll be using a set level data table as opposed to sample level data table. So the major takeaways here is Terra is a web application that connects users to bioinformatics workflows and cloud computing resources through a graphical user interface. And workflows are important from Docsto. and compute and data storage hosted on the Google Cloud platform. And data on Terra is organized into Terra data tables. And we've talked about, two different kinds of Terra data tables, sample level as well as set level. And we'll talk about how, the data in the Terra data table can be accessed using this particular notation, and how to select and define the inputs that go into running our workflow. So are there any questions up to that point? And if I'm moving too fast, please let me know. And yeah. But also remember that we'll have another session on Thursday where we can go and revisit all these that you're talking about today. But still feel free to just ask a question so that I can help clarify anything. Okay, so yes, Rotimi.
Thank you, James. I don't think you're working too fast. I mean, for me at least. So I wanted to ask, who is responsible for creating workflows? Is it what you do at your back end of the server, or what individuals who want to host some kind of workflows will do? I hope my question is clear.
Yes, your question is pretty clear. So, I mean, like I showed on if I can just go back, so different organizations develop these workflows. For example, Theiagen have their own workflows, the Broad Institute have their own workflows. And then once they've written these workflows, they package them. Of course, the institution, depending on what their interests are or their research interest. You as an individual as well, you can also write your own workflow, for example, in WDL, have it on GitHub, package it onto a Docker container, host it on Dockstore, and then you can pull it onto Terra, or you can have others pull it onto Terra as well. So yes, bioinformaticians typically do write the workflows, and then they push them to be hosted on Dockstore, and then they can be pulled onto Terra quite easily. Rotimi, does that answer your question?
Perfectly, thank you.
All right, good. Okay, so quickly moving to this stage. So once data has been uploaded and organized into a Terra data table, we can analyze it with a workflow. So the workflow receives inputs from a Terra data table. And then once the data has been processed, depending on the workflow, it will populate those outputs onto a Terra data table. Now, we want to get our data onto the workspaces that you assigned using two methods. So one, we want to use SRA fetch. And what SRA fetch does is to fetch raw reads from the SRA, which is the short read archive by NCBI, into your Terra workspace. And this means that, if you have your reads already submitted to SRA, then you can have those pulled onto your workspace without having to actually physically upload the FASTQ files if you had them like on your computer or on an external drive. And as you might be aware, the short reads will typically have prefixes like SRR, ERR, DRR, again, depending on, whether, the specific database, whether it's the Japanese one, the European one, or the one that's based in the US. And every data, of course, will have an accession scheme. So second letter is always R. The last letter denotes the hierarchical level. So you have the study, you have the sample, you have the experiment. So you'll always see this in the accession scheme, like whether it's SRP, SRS, SRX, SRR. So if you've ever submitted these row reads, or even like looked at reads and projects within SRA, this will be something that's quite familiar. For our context, we always want the run, which has the row reads, and these are the accessions that we'll import onto these workspaces. So one, we'll demo how to run the SRA fetch workflow. And I want us to do this because it can be running in the background as we also look at the alternative manual upload process. So, so here what we'll do is for the SRS fetch, we'll have to upload a TSV file with a sample name and an SRR accession for each sample in Terra format. We'll see what that means. And then import the SRA of fetch workflow from Docstore. We have already done that, so we don't have to redo that. We'll see how we can do this when we do RC typing, which is not available yet on your workspace, and then run the SRA fetch workflow. So the TSV file, so this is a typical tab delimited file, or also similar to like a .txt file. For the Terra format, there are critical elements that you'll want to note here. One of these is you'll see on the first column, we have the column header starting with the word entity, and then has a name there. For example, in this case, Bioinformatics analyst _ training _ sample _ ID. So the thing you'll notice here is that there is no white space between the names. So entity is always a must. So that column identifies the data table. So that will be the name of the data table that you'll be uploading onto Terra. You can name it anything, whatever you want. The only thing, as I've mentioned, is there must be no spaces in between, whatever names you put. And the end of that name has to end with _ID. And that is how the Terra acknowledges that that is your sample name column. So it's sort of like it's the key. If you understand the concept of databases, the key value pairs, so this is sort of like a means of identifying that this is the key to your data table, which will be your sample IDs. And then on the second column here, we have a column called SRR accession. And then, you can have as many different columns as you need, depending on, additional metadata that you might want to upload together with your sample list. Again, remember, you could even decide that, your sample column could just even be just the accession, so that you don't have sample 1, 2, 3, 4, 5, 6. But again, this is something that, just to think about, because, for example, if you're dealing with patient samples, sometimes you might not want to deal, for example, have the actual patient sample IDs, but, have some auto-generated sample IDs that you can work back to the individual patient IDs. So, yes, Rotimi.
Sorry, James, again, just the last page. I was thinking if you use the second column now, if you had used SRA for sequence read archive _ session, I mean, it should make sense to anybody, because if you use SRR, you do have DRR, you have ERR too. So I was thinking, is it possible for us to use SRA _ session? I mean, just my opinion.
Absolutely. Yeah. So remember, the name of the column does not matter. But I absolutely agree with you that, you should have columns that are sort of like self-identifying or self-explanatory. that if you, especially, in lab workspaces, if your fellow colleague goes in there, they can quickly figure out what that column is for. Perfect. I definitely agree with you. So if you've prepared a TSV file like that, then all you need to do is come to the data tab, then click on the import data button here or tile, and then you'll see a drop down with multiple options. One is to upload TSV. The other one is Open Data Uploader, which we'll use for the manual upload. And also to add reference data, which we'll not talk about in this particular training, but you can typically have reference data that can be used across different workspaces as well. And once you import, you click on import TSV, you'll get a pop-up, where you can upload that TSV or text file from your computer, or you can just drag and drop it. And once you do that, you'll see that, the name of the table will pop up on the left here, and with the columns that, are in that particular TSV file. Okay, let me just go, actually, probably it will be easier. Let me just continue this. Once you uploaded your data, the next step will be to undertake the analysis. So I'll switch back to the training resources page. You'll notice that on the right here at the top of the page, we have this category called training datasets. So we have data that you need to download to your local computer. So there's a reads for upload.zip. So this is a zip file that has some raw read FASTQ files. And then we have the training underscore data manual TSV. So this is the accompanying table for that. We also have an option typically for participants that are in places where data might be a problem. So in this case, we have stored the data in on the cloud, on a bucket somewhere. Then what we'll be importing is actually the links to that particular bucket, which will get the data onto your workspace instead. And then we also have data that we need to fetch from SRA. So if I can just quickly shift here. So here I have data to fetch, that's what we call it. So I'll just open it with Excel here. Okay. So you see in my case, I have only a single column. And if I can just zoom here a little bit. As I said, you can see we started with entity, and then we have our column there, and then I'm calling this training underscore data, and I'm ending it with the underscore ID just as an identifier. And I have these SRA sessions here. So I don't have a separate column here. But then again, remember, you can have as many columns as you need, depending on what you need that data for. So I'll just close this. Again, what is important is ensure that this is tab delimited file, which means, it's separated by tabs, not, for example, CSV or even like .xlsx format. So going back to my Terra workspace here. So I'll come to the data tab. I'll click on import data. Then I'll click on upload TSV. So you can drag drop, or I can just click here, and then this pop-up will come up. And then this is the data table that I need. I click open. And once it's listed here as the selected file, you can see data to fetch.tsv, which is the name of the file. The other alternative is you can also copy paste if you prefer that option as well. You can just copy and paste it there. And then I'll say start import job. And on a flash, you can see that now I have a data table under my tables tab here. And you'll see that we have the list of those samples or accession numbers here. And as you'll see, this is a sample level data table where each row represents a single sample. Now, what we want to do is undertake or run as the SRA fetch workflow. So what the workflow does is to use these accession numbers to pull the first Q reads onto our workspace, which we'll run through Theiagen later this week. So I'll go to my workflows tab. We already have the SRA fetch workflow imported here. So I'll just click on it. So what you'll notice here is for bioinformatics, the versions of the workflows are quite important. So, just like in the lab, you always note, for example, which batch of reagents that you're using, etc., or the barcode. It's the same thing with this workflow. So if I look through this drop down, you'll see that there are so many versions of this particular workflow. It's always advised to use the latest stable version. So up here, you'll see that, there are these different versions that have names. So these ones are often versions that are still in development. So they're still under testing, so you don't want to use those ones. You always want to use the latest one. You can see we have 3.0.0 as well as 3.0.1. So once you've selected that particular version, so here we have options like run workflow with inputs defined by file paths, or run workflows with inputs defined by data table. We want to run our workflows with inputs that are coming from the data table that we just imported. So once I've selected that, I just click here, and you can see that already, it's also preempting that, I'll have a set level data table that goes along with my sample level data table here. So I'll just select my training data sample level data table, because remember, we are going to fetch each sample reads independently. Then I'll click here on select data. So if you typically want to select all the data on a data table, then you normally want to click this small triangular button here and then go with the option of all. You can typically, as you know, in most web pages, when you click this button here, it should be able to select all. But there is a limitation on Terra whereby if you have a table that has more than 100 items, it will select the first 100 and then totally forget about whatever it is on the next page. So if you mean to select everything on your data table, then always go for the all option. So once you've selected data table and the samples there, you'll see that it even tells you that this selected data will be saved as a new training data set called that. And I'll just click OK. And here you'll see that now 40 samples that have been selected. So this also helps you have an idea of actually how many samples you've selected and if that is the number that you wanted to select. Now, there are a couple of different options here. So typically, the most important ones are like use this call caching. Of course, you can hover your mouse here. It will give you an idea of, or just click on it, what that option is. So this typically means that if the workflow has been run before and it failed, let's say at a particular stage, it might not need to start from the very beginning, but at that stage where that particular workflow failed. So helping you with some compute time and of course money as well. In this case, we won't select any of those other options. Now we have this section here. So this is where we input the columns where we want the different variables to take input onto the workflow. The thing that you'll notice here is under this variable column and also type as well, you'll see that this one is in bold, while these ones are not. So the ones in bold simply indicate that that column is required. And of course, if these cells are empty, you'll also see that written as required and these ones being optional. So required means the workflow will not run without a value in that specific value column or cell. So to get, I think the thing I'll do, let me just open this as a new tab because I want to be able to jump back onto this table here. So you'll see that, this column is called training underscore data underscore ID. So that's where we are going to get our accession numbers. And typically Terra will preempt so to select a given column on the data table that you've selected here, the notion that we use is a prefix called this dot, indicating that I want to select the column from this particular table, and the column is called training underscore data underscore ID. So if I just click on that there, so that will be filled. I don't need to fill this time any of these other options. So remember, these are optional. And then I'll go to the Outputs tab. Then what I'll need to do here is I'll click on Use Defaults, which means I'm telling the workflow to use the default outputs for that specific workflow. So once I've done that, I'll just hit here Save. And then once it's saved, you can see that now the workflow is ready to launch. So I can click here, Launch. Then this pop-up comes up that says Confirm Launch. Typically, you have this window where you can, write something about, the particular workflow run that you're launching. Again, going back to the lab, for example, where you'd say batch 7 or batch 10, or like, it could be because of the workflow, you're also trying different parameters. So it could be like, oh, I'm testing this workflow with this quality score or something. So I can say, for example, my first SRA fetch. And I'll just click launch. And then you'll have a few messages there, and then it will quickly take you to the submission history tab. And you can see here, it's on the workflow showing you what has been launched, what has been queued. On the input, it will, you can see the input options that you made and the outputs. So these again, remember, because Terra is table-based, so these are the columns that we expect some values to be populated onto. So if I just click on the submission column again, you'll see that it will be listing the workflow that we've just launched, the number of workflows here. So ideally, these are the number of samples. The current status is that it's been submitted, and the time, the submission ID, and the comment that we made there. Don't worry. I mean, if I click back and you needed to change this comment, you can edit it back, and you can, for example, say 17th June 2025. I mean, if you needed to add something, you can still add it later. So no worries at all. Okay, so that will be running in the background. You can see that this has changed to submitted now, and that will be proceeding as we go ahead. Are there any specific questions on running the SRF fetch workflow? Okay, cool. So we said that, let me just ensure that the data here is, so we downloaded that reads.zip file. I just wanted to unzip it and have those reads ready. So you should be able to see 2 samples And you have the forward and the reverse reads for each of those samples. And for this specific reads, again, I'll just open this, our data table that we'll upload at some point to Terra. Again, you'll see that, we have the data table called training_data_ID. And therefore, this means that, it has the same name as the data table for which you are doing SRE fetch. And that implies that, that data will be added, this data for these two samples will be added onto that data table. You can see we have columns read 1, read 2. And if I go back here onto our workflow that is currently running, you'll see that in our outputs, we also do have two columns, read 1 and read 2. So that means that, again, our data table will populate onto an existing table where these two columns are present. So we'll go here. So now we want to do that manual process because this might happen. You've done your sequencing, you've gotten data out of your Illumina machine, for example. You have that data in a drive. And then you want to upload those onto Terra. The third option that probably will not cover this time, but is also available, especially if you use base space. There's also a workflow called base space fetch that has also been developed by Theiagen. And that means that, you don't have to download your raw reads from base space onto your local hard drive and then upload onto Theiagen. You can actually pull that data from your base space account onto Terra directly, but it involves a bit of a process where, like, you have to give Terra certain permissions to be able to access your base space account. So that, if you're interested in that option, then definitely let us know. We can explore how we can help you with that process as well. Okay. So we'll go here to import data. In this case, we are going to use the option Open Data Uploader. So this time we are not going to the upload TSV. So in our Open Data Uploader, it's asking me to select the workspace where I want to do the data upload. So it's going to be my demo workspace. I mean, for you, you'll see only one workspace that you have access to. For me, I have different options because I have, access to different workspaces. So in this case, I'll select this demo workspace, just ensuring that it's the same name. And then it asks me to select a collection. So think of it like for every batch of data that you upload, you might want to think of it either as a collection, or if you have a collection, you can just add on to it. So I want to create a new collection, and I want to call it, let me just call it JIDC training data. Again, you're noticing that I don't have spaces in between my names. Again, this is just a good bioinformatics practice because some tools typically have issues with spaces in between names. And then I'll create that collection. And then the next step is now to upload my data files. So I'll click here, upload, and I'll go here to my reads. Then I'll just select 4. And then you can see that, it's undertaking the uploads, upload in progress. This step is typically dependent on your internet speed. If it is slow, it can take a bit of time. If it is fast, obviously, it will take the shortest time possible. So you can see we're on to the second file of 4. So it should be done pretty soon. Any questions in the meantime as we wait? Okay. It's good. There are no questions. Okay, so going back just to our SRFH workflow that is running, you can see that there is a green check mark. Remember, we had 40 samples, so you can see when I hover my mouse onto this area here under the status column, it's saying that 15 are done. Fortunately, there is none under the red triangle, which means they failed, and 25 are still getting processed. And should I go actually to my data tab and to this specific data training table, you'll see that the 15 that have already been processed, you can see that now we have additional columns on this data table. And particularly of interest is read 1 and read 2 columns, which, it has fetched this data from SRA, and the rest will also be populated as they're processed. Okay, so we are on to the last sample there. So yeah, this SRFH is also pretty quick here. Okay, fantastic. So you can see that, our 4 files have been uploaded. And actually, if you hover your mouse over this particular file, you remember I told you that these are stored in a Google Cloud bucket. You can also just download it back onto your local machine if at some point you need to do that. And here it even shows you how you can do that on the command line. But in a way, this also gives you the path to where that particular file is, and you can see the specific bucket here, which the data is stored. All right, so once we've uploaded our files, I'll just click next here. And then the next step is to upload my metadata file. Of course, it can also do auto-generate this for you. That is, if you didn't have one pre-prepared, again, just basically depending on whatever is on that data table. Anyway, in our case, we have our TSV file here that I want to upload. Just before I do that, coming back here, you'll notice that these names that I have under read 1 and read 2 have the exact file names for the files that we just uploaded. That is quite important because that is how the Terra is able to link the data table to that specific file that you just uploaded in that upload process. If the name is different, then it will not link. So we'll take that file, upload it, and you can see that it's linked our file with the specific files that we uploaded. But there's also a warning message here that says that this workspace already includes a table with this name. If any new rows have the same training data ID as the existing, then the data in that row will be updated with new values. So essentially saying, if you uploaded a new sample, if you uploaded data with the same sample name as a sample that's already on that table, then it will be overwritten with the new data. In our case, we don't have that, so we'll just say update table. And then once it's been done, you have the option of view training data table in the workspace. So you can see that now we've added 2 more samples. So now we have 42 samples instead of the 40. Okay. So if I go back here, okay, there's 1 that has failed. We have 8 pending there. So again, this is showing that some have succeeded, 1 has failed, and we can we'll go over these, like how to troubleshoot why a given sample has failed, etc. So just go back to my slide set. So we have 3 minutes to the top of the hour. So we've gone through how to upload or get data onto Terra using a manual process. And we've also looked at how to import that data from another repository, in this case, the Short Read Archive or SRA. So on Thursday, we'll be able to go over this again. So what I would want you to do between then is try go to your training data page, download this data, try to follow the steps, and then upload data onto your workspace manually and also through the SRA fetch. Of course, I gave the option that, if you have internet issues, then we have this option. So you can see here, instead of my file, let me just expand this. Instead of my file just reading this _fastq.gz, you'll see that here you have a path that goes to the a bucket that hosts a file similar to the one that we upload in that particular Google Cloud bucket. So if you have internet connectivity issues, of course, you can use this data table instead. It should still be able to give you those two samples or add those two samples onto your workspace. But I would encourage you to try that manual upload process just to be familiar with it. So I'll avail the slides here under week 1, the ones we've walked through today, just so that you can be able to undertake the exercise by yourself. And also the recording will be added onto this section here for the recording. And please do reach out if you get stuck at any point, and write me an email, and I'll be able to respond and see how I can help. All right, 1 minute to the top of the hour. Are there any questions? Okay, so I don't see any hands. So Thursday will be more interactive. So, you can get to do this during the 1 hour, or we can get to address any additional questions there. So it's meant to be, less talk and more discussion. So please feel free to join then, and I'll be able to address any questions that you might have. Otherwise, thank you for attending this session and hope to see you on Thursday.
Thank you, James. See you on Thursday.
Yeah.
Thank you.
Cheers.
That's fine.
All right, I've just stopped the recording. In case anybody has any questions that they did not want to be on record, so you can squeeze in a minute or so. Otherwise, we can also just end the call. All right. Okay. See you all on Thursday, same time.
Thanks, James. See you.
See you, Jen.